Hello everyone, welcome to Modern Microbiology course with อาจารย์ลัคนากันหายุวะ Wingfield. Today we are going to learn about the molecular identification of fungi. Identification of fungi to species levels is paramount in both basic application and applied application. For basic applications such as ecology and taxonomy study, and for applied applications such as genomics and bioprospecting study in scientific research. This is especially true for natural products researchers working with fungi as a source of bioactive secondary metabolites. Scientific names are crucial in communicating information about fungi, enabling researchers to identify other closely related species to better predict evolution of chemical genes clusters, or to prioritize taxonomically related strains when a productive strain may attenuate production of key bioactive compounds. More importantly, taxonomic identification of fungi is essential if industrial, agrochemical, or pharmaceutical products are to be derived from a fungal strain. Mycologists have traditionally used morphology, like phenotypic characters, such as spore-producing structures formed as a result of asexual or sexual reproduction, as the sole means for identifying fungal species, and even today it is still adopted as a means of species identification within a mycological community. Use of morphology in fungal species identification is very important to understand the evolution of morphological characters. However, morphological approaches to fungal systemics, although routinely used in fungal taxonomic studies for classification of fungi at the ordinal or familial level, may not always perform well for lower levels like species levels and classification. Moreover, identifying fungi based on morphology alone can be challenging, especially when non-experts are dealing with cultures of fungi, since there are a limited number of morphological characters that can be used for identification. For example, endosymbiotic fungal strains such as endophytic fungi, which are routinely used for isolating secondary metabolites, do not always sporulate in culture, thereby providing no phenotypic characters for which to identify them based on morphology. For the fungi that do not sporulate in culture, the asexual structures such as conidia, spore shapes and size, can often show highly plastic characters, making identification challenging. As I told you earlier, there are several problems of using morphology alone in identification of fungi to the level of species. These problems are, for identification, using morphological character, you will be able to identify the higher level like order or family or genus level, but not into the species levels. You will also need an expert for identification of fungi. And for the fungi that are non-sporulating fungi, you will not be able to identify them because you cannot find the reproductive structure that help you to identify. And especially there are so many variations of fungal characters which is hinder the characterization of the fungi. And lastly, the most important thing is a morphological identification is very time-consuming and laborious, which for performing a fungal identification using the morphological study alone is not important. That's why using the molecular technology is involved, and they are very important nowadays. There are so many methods that you can use to identify fungi. For example, fluorescence in situ hybridization, DNA array hybridization, multiplex tandem, PCR, mass spectrometry, multitoph, sequencing and pyrosequencing, and DNA barcoding and DNA taxonomy using the sequencing technology, which I'm going to introduce you in the next topic. Before we move on to the identification of fungi using the molecular technique, let's see the overview of how DNA barcoding and DNA taxonomy are generated. DNA barcoding involves the use of ITS region for fungal identification by comparing your unknown against the sequence database. And then you will need to identify based on the sequence similarity of your query sequence to the database. And for DNA taxonomy, this method can be used one or multiple gene for identification by performing the sequence alignment, and then you will need to construct the tree using a variety of tree building tools. And these are the publications that I follow, and I'm using some of the information from this publication to teach you to generate the DNA barcoding and DNA taxonomy. The publication entitled Fungal Identification Using Molecular Tools, a Primer for Natural Products Research and Community. And here I'm showing you the abstract. If you are interested to read more in detail about the what this publication talked about, you can download it. It's the open access for everyone. Let's move on to the first topic. The key that I would like you to know is the 3 nuclear ribosomal genes that most commonly used in fungal identification. The fungal DNA sequences generated with fungal nuclear ribosomal operon primers for the last subunit, or NRLSU, excuse me, 26S or 28S, small subunits, or NRSSU or 18S SSU, and the entire internal transcribed spacer region, which are ITS1, ITS2, and 5.8S. And these regions usher the use era of molecular phylogenetic sequence identification in the kingdom of fungi. Different rates of evolution resulting in varying level of genetic variation have been observed for these 3 separate regions, with SSU evolving the slowest, thus possessing the lowest amount of variation among taxa, while the ITS evolves the fastest and exhibits the highest variation. If researcher is interested in the phylogenetic placement of a fungus at a higher taxonomic level, like family, order, class, and phyla, the SSU can be amplified and sequenced using the primer combination of the NS1 and NS4, and you can also use the combination of other primers, and here are the example of primer sequences. When you use the NS1 and NS4, it will generate the PCR product of about 1,200 base pair. And for alternatively, if the identification needs to be made at the intermediate taxonomic level, like family or general level, then the user can amplify and sequence the LSU using the primer combination of the LROR and LR6, which is here. And you can also select other primers that will generate the region of these LSU regions. For using the LROR and LR6, it will generate the PCR product of about 1,200 base pairs. As I mentioned earlier, for the SSU, this region evolved very slowly, so it is useful for higher level like family and order phylogeny. And for the LSU, this region evolved slower than the ITS but faster than the SSU, so it is useful for intermediate level like genus level phylogeny, which makes up the largest database in the International Sequence Database, along with the ITS region for fungi. For species level identification, the ITS is the most useful as it is the fastest evolving portion of the ribosomal RNA cistron. Due to its ease in amplification, widespread use, and appropriately large barcode gap, the ITS was chosen as the official barcode for fungi by a consortium of mycologists. Hence, for natural products research, the first two domains of the LSU gene and the entire ITS region should be sequenced due to their high prevalence in fungal taxonomy and systematics. And here are the regions of the ITS1 and ITS2. For amplification of the ITS1, you can either select the primer ITS1F to the LR3, or ITS5 to the LR3, or ITS1 to the LR3. And the entire ITS and the first two domains of the LSU will generate about 1,100 to 1,250 base pair of the PCR product. And here are the example of other PCR primer sequences that you can select. For the amplification of the ITS2 region, you can also use the ITS1F or to the ITS4 or ITS5 to the ITS4 and ITS1 to the ITS4. And the entire ITS region will generate the PCR product of about 550 to 600 base pairs. For the ITS regions, it evolved very fast, so this region is very useful for lower level or species phylogeny and DNA barcoding marker for species identification of fungi. And even though the ITS region performs well as a suitable fungal barcoding marker, it has been subjected to a debate. The ITS region does not work well in some highly species genera, such as Aspergillus, Cladosporium, Fusarium, Penicillium, and Trichoderma, as these taxa have narrow or no barcode gaps in their ITS region. From this reason, there are another alternative use for the identification of fungi by using the various protein coding genes, which is used to augment or supplant ITS in species level identification of certain fungal groups. As I mentioned earlier that ITS alone are not sufficient, so it is very necessary to use the protein coding genes to get more precise identification, especially for the fungi in the genus Aspergillus, Penicillium, and Trichoderma. Protein coding genes are utilized for species identification via barcoding due to the presence of the intron regions, which sometimes evolve at a faster rate compared to the ITS, and are employed in phylogenetic analysis due to their better resolution at higher taxonomic level compared to the rRNA genes. Moreover, these genes allow for an easy recognition of homology and convergence, as they are believed to occur as a single copy in fungi, are less variable in their length since they accumulate fewer mutations in their exons, and are easier to align over ribosomal RNA genes as they certain less ambiguity due to codon constraints. Protein coding genes in fungal systematics have been used widely for constructing molecular phylogenies to aid in an identification and classification of taxa. These efforts among fungal systematists leads to the way to a more stable classification of the fungal kingdom and impart knowledge regarding gaps in an understanding of evolutionary relationships among fungi. Among protein coding markers, the largest or RPB1 and the second largest or RPB2 subunits of RNA polymerase translation elongation factor 1 alpha or TEF1 and beta tubulin or TUB2 and BEN-A have been mostly commonly used for inferring the phylogenetic relationships among fungi. More recently, the mini-chromosome maintenance protein or MCM7 has shown promise as a new marker for inferring both higher and lower level phylogenetic relationships. After you get the sequence of all those regions that it can be used for the fungal identification, the most important thing that you need to think about is how to use the NCBI-BLAST search for DNA barcoding. Each ITS sequence fragment is subjected to an individual basic local alignment search to or BLAST in GenBank to verify identity. BLAST search is usually employed using nucleotide collection or BLAST-N. For BLAST search, it is reasonable to start with more than 80% QLI coverage and more than 97 to 100% sequence similarity, for example up to 3% sequence divergence for assigning a species name. However, there are some limitations of using GenBank BLAST search. It has been advised that BLAST search identification against GenBank should be made with caution, as approximately 27% of GenBank fungal ITS sequences were submitted with insufficient taxonomic identification. In addition, about 20% of fungal sequences in GenBank can be incorrectly annotated, but it will vary greatly according to a taxonomic group. And here are the examples of the result when you get the result from the NCBI-BLAST. There are so many columns, but there are some columns that you need to focus on, which is the first one, which is showing the sequence that hits your query from the database, and all these scores indicates the degree of similarity between the query sequence of your unknown and the hits from the database. So for the query coverage, the higher is the better. And also with the sequence similarity, which is the identity here, the higher is the better as well. And one thing that you will need to consider is the E-value, which are an indicator of the likelihood that a given match was generated randomly. So the lower of the E-value is the better. For this example, you will see that the percentage of the query coverage is about 100% for all hits, and for the identity it's more than 97%. So I can conclude that the unknown mushroom of mine is the Lentinula iodes. Apart from the gene that you will need to consider when you have to do the identification, one thing that you will need to consider is the method that you choose to use for the construction of phylogenetic tree from DNA sequences, which will facilitate fungal species identification. Like DNA barcoding, DNA taxonomy is based on the premise of using the genetic variation inherent among sequences of different individuals to identify taxa. However, the difference between these two methods is that the latter, an unknown, is identified based on a phylogenetic hypothesis, that advocating an evolutionary perspective for recognizing fungal species and utilizing an approach with predictive value. DNA taxonomy for a particular group of fungi may be based on one or more regions of protein coding or ribosomal DNA, and can be derived from the phylogenetic methods using any gene region individually or in combination, while DNA-based taxonomy using phylogenetic theory may not always help with identifying the exact species of the fungus. It will certainly help by placing an unknown species within a phylogenetic clade or group, so providing a putative species identification. So, to example a phylogenetic tree, traditional approaches such as neighbor joining algorithm, which is the distance-based method, and the parsimony, which is based on the insertion that the base phylogenetic hypothesis requires the smallest number of evolution steps, were used previously. However, both of these have been supplanted by faster model-based methods such as the maximum likelihood and Bayesian inference. This is largely because distance-based and parsimony-based methods do not perform well with molecular sequences that are evolving at a different rate. And there are several tools that you can use to construct the tree. For example, the cluster omega, which is easy to use, and make a program, phylip, and map. And here are the examples. Before I perform the tree construction, I will need to select the best hits against my unknown, and then you will have to retrieve the sequence of the gene or region that you are interested, and then transform it into the FASTA format before submitting the sequences into the program. And here are the overview of how will you perform the fungal identification using the DNA barcoding and DNA taxonomy. And here are the example of the DNA barcoding, which is in the panel A. The sequence data using the ITS or other taxon specific genes can be used to obtain directly from the fungal fruiting body. In this case is the mushroom, and then a BLAST search is conducted in the INSD database such as the GenBank or others specialized database. And then identification is based on an overall sequence similarity. For example, 79 to 100% sequence similarity with other reference sequences in the database, thereby providing an identification of an unknown species. The example is for the mushroom Lentinula eodes, which was recently published in the. And for the DNA taxonomy at the panel B, after BLAST search, the most homologous sequences, usually at the top 50 to 100 sequences, are downloaded from the database and then incorporated into a multiple sequence alignment and phylogenetic tree building methods such as maximum likelihood and Bayesian inferences are applied. Using these methods, the unknown sequence is identified within the evolutionary framework, and the example is from the research from this publication is the mushroom G77 strain, which was published previously in this journal, are identified as the Aspergillus isuke. And I'm going to tell you in the detail of each panel. For DNA barcoding, if you would like to identify your unknown fungal sample or specimens, the first thing is you will need to extract the DNA to get the genomic material. In this case, this publication gives an example of the unknown mushroom, and they are going to perform the DNA barcoding using either the ITS sequence or other DNA marker. After you do PCR amplification, you will get the region of the ITS sequence in this case, and then you will need to do the BLAST search in the International Sequence Database or other specialized DNA databases. And then after you get the result, you will have to use a cutoff proxy to be considered the same species that the ITS should have more than 97% similarity with more than 80% coverage. And here are the query sequence matches database sequence, and then you will get the result. After you getting the result, you will need to consider the scores showing in here. For example, the query coverage, which is shown like mostly 100%, and sequence similarity or identity is 99%. As I told you earlier, that E-value is very important. In this case, the E-value is only 0, so the lower is the better. And for the query coverage and identity, the higher is the better. So from this result or is line, you will see that for this example of this unknown is the mushroom, we can identify it into the species level as the Lentinula eodes. To perform the DNA taxonomy, in this case, they are using unknown filamentous fungi, which is the strain G77, is the unknown culture. First, the DNA sequence database, the obtained from the culture is BLAST. After you get the sequence information, you will need to do the BLAST search in the INSD, which published and authentic sequences includes the X-type sequences from holotype of the RepSeq database. After you get all this information, after you BLAST, you will get the result like this, which is similar to the first example. In this case, they are showing mostly the genus in here is the Aspergillus, and there are variety, a diverse species here. So we cannot make a conclusion what is the species names of these Aspergillus. By looking at the query coverage, it's mostly like more than 80%, so we cannot make the conclusion. E-value and identity is very high. So what we need to do next is we select the top 50 or 100 BLAST search matches and then align them using multiple sequence alignment software. After you get that, you generate a tree by doing tree construction based method using the program that I mentioned earlier, and then you will get the phylogram like this. For this example, which is very small, the G77 is here, which is in group with the Aspergillus iisuke. So the unknown strain of the G77 is placed with a known species from a type strain. In this case, is the Aspergillus iisuke. Numbers refer to the bootstrap support here, and the phylogram of the most likely tree from using the RAXML analysis of 60 taxa based on the complete ITS and partial region of the 28 H-last subunit or nrDNA of the 1000 base pair. Let's have a look closer at the phylogram. That you will see this is the bootstrap number, which is quite reliable. And here is your unknown, which is G77 here. They are grouped in the same node with the Aspergillus iisuke. So in this case, you can make the conclusion that your unknown could be the same species as the Aspergillus iisuke, section FLOWCHART. And before we finish this topic, I would like to make a summarize by showing the flowchart, which is generated by the publisher. Here is the flowchart of the fungal for the fungal identification using the molecular phylogenetic analysis from the first step to the last step. So if you have your unknown culture and you would like to identify them using the molecular method, you will have to extract the genomic DNA from culture and then perform PCR amplification using whatever you've got in your lab by selecting gene or region that you are interested and you would like to do. For example, you can combine the ITS1 with the ITS4 or other ITS region. And you can also combine the ITS primer with the LSU region, so you will generate quite a variety of PCR products here. Then you send your sequence for Sanger sequencing, and after you retrieve your sequence information, you submit your sequence by doing the sequence BLAST search into the International Sequence Database. And then you will have to use the search marker for phylogenetic analysis. For example, the use of RepSec loci in the INS database, GenBank, EMBL, or DDJB. They are very useful and have quite the same result. And after you get the information, you will need to conduct multiple sequence alignments. And also you will need to remove ambiguous regions with any program like gBlocks and perform maximum likelihood analysis and then run bootstrap of 1000 replicates, and then you will construct the tree after that. After you get the tree phylogram, you can make a rough conclusion what your unknown is about. And the most important thing is you will also need to perform morphological analysis when possible to confirm the characteristic of your unknown. And if you cannot make the conclusion, you can go back by changing the gene for fungal identification, like using the protein coding gene like RPB1, RPB2, beta-tubulin 2, and MCM, something like that. However, you will also have to realize that ribosomal genes may not always provide an accurate species identification by using the ITS. So, the user are encouraged to sequence protein coding gene, which may provide better resolving power for certain lineages of fungi. And lastly, I would like to make the conclusion or summarize or remember concept for you to remember, which this publication has divided the concept and conclusion into four parts. The first part is whenever possible, identification of fungi should be made using a combination of microscopic morphological, cultural, and molecular characters, and the entire ITS region alone or in combination with the first two domains of the LSU and one or more protein coding genes in specific cases should always be compared with authenticated published sequences. Such information would appropriate for the supporting information. For manuscripts in the chemical literature, one should strive for at least genus or family level identification. If a fungus garners enough interest for further study, then it may be possible to follow up with species level identification in the future using methods outlined in this publication. Second, the ITS region works well in most cases, but should be used with caution, especially when the user is employing only the GenBank-BLAST search for identification. It is recommended that the user confirm GenBank identifications against curated sequence data, as these data can be more reliable. For cases where the ITS region is not ideal, such as case 1, like when there is not sufficient resolution of species identification in an Aspergillus, Fusarium, Penicillium, and Trichoderma, or another case when one is working with multiple morphologically similar species which may exhibit rampant cryptic speciation. The user should analyze other protein coding gene regions as well. Third, one should conduct phylogenetic analysis with nuclear liposomal genes first, since more of the data are available for comparison in public databases of the ribosomal gene. Once the user is able to narrow in on a taxonomic group or lineage, or if ribosomal genes are inconclusive, protein coding data can be utilized in combination with ribosomal genes or separately for finer scale species level identification. DNA taxonomy employing phylogenetic analysis is useful for placing an unknown sequence into an existing classification in an evolutionary framework. And the last conclusion, it is extremely important to use sequences that are published for comparisons and, where possible, to reference an original source of the sequences. The user must always deposit the sequence data to GenBank, and often after the manuscript in which the sequences are reported and published inform GenBank about the pagination. This is often not done by many researchers, which results in many unpublished and unreliable sequences in the GenBank. So, in summary, both basic applied fungal research would benefit tremendously from DNA barcoding, especially when morphological examination is not possible in sterile cultures or when morphological data are inconclusive. And I would like to end the lesson today. Thank you everyone for attending the class. Have a pleasant day. Thank you and bye-bye.